Okay, so let's take a look at chromosomal mutations. That's the last part of this chapter. These are chromosomal mutations are changes that occur in either the number of chromosomes or the chromosomal structure. These are detected by what is known as a karyotype. And so this is showing a karyotype. This is an arrangement of chromosomes based upon their size and centromere position. So as I mentioned, it's used to detect chromosomal abnormalities. So what we can do, in a karyotype, we match up chromosome number 1 from your mom with chromosome number 1 from your dad, chromosome number 2 from your mom with chromosome 2 from your dad, and so on, and then we have the sex chromosomes there at the end. Okay? So oftentimes this is done before somebody is born to see if they might be born with any chromosomal abnormalities, and these are two of the main ways that this is done. One is through an amniocentesis. And so here we have this sac around us called the amnion. And so using an ultrasound, we stick a needle in and pull some of that fluid out, and that fluid will have fetal cells. Another way is through a chorionic villi sampling. And here we take a little piece of the placenta. This can be done earlier on in development. All right? And then you do a karyotype of this. This can also be done with blood cells as well. All right? So what we're looking for are either changes in chromosomal number or changes in chromosomal structure. So let's look at changes in chromosomal number. These are caused by this process called non-disjunction. And non-disjunction is where the chromosomes fail to separate during meiosis. So what we're supposed to see is disjunction. So in disjunction, the chromosomes separate, like what we see here and what we see here. Non-disjunction is where they don't separate. All right, so two ways that this can occur. One way is that homologous chromosomes fail to separate during meiosis 1. And so what you get here is you get 2 cells with 1 extra chromosome, 2 cells missing a chromosome. The other way is where the chromosomes fail to separate during meiosis 2. So the sister chromatids fail to separate, they both go to the same cell. So here you have 2 normal cells, and so 2 normal gametes. Here, 1 gamete with 1 extra chromosome, 1 gamete missing a chromosome. So the normal chromosome set is called a euploid. So, for humans, that would be 46. All right? So in one kind of situation is called polyploidy. And this is a cell with extra sets of chromosomes. So instead of non-disjunction occurring with 1 chromosome here, it's going to occur with them all. So in this case, you produce diploid gametes. And those diploid gametes aren't going to match up with haploid gametes to produce viable offspring. They have to match up with another diploid gamete. And in most cases with animals, this is a lethal condition. But with plants, this has actually led to different species of plants. Fully 30% of all flowering plants are polyploids, having several sets of chromosomes. So such things as chrysanthemums and watermelon and even bread wheat are all polyploids. Now let's look at aneuploid. Aneuploid is a cell with 1 or more extra or missing chromosomes. So instead of having 46, you could have 45 or 47. Now, when it comes to a monosomy, that's a cell with 1 missing chromosome. With humans, if this is autosomal, that is lethal. It does not develop. All right? So if you're missing 1 chromosome in one of these autosomes, so one of these guys here, you don't develop properly. If it's in a sex chromosome, so if it's in a sex chromosome here, if you just get a Y chromosome, that doesn't develop either. But if you get just an X chromosome, well, that will develop. And this is known as Turner syndrome. So this is a person who's usually short in stature, might have some mental deficiencies, and they're typically infertile. The other case is called a trisomy. And a trisomy is where you get 1 extra chromosome. So this is a cell with 1 extra chromosome. All right, so what we see here. Now, if this is autosomal, one of these non-sex chromosomes, this is mostly lethal. Doesn't develop properly. And one of the problems there is some of these, like 13, 17, 18, and 19, can actually go into late term pregnancies. So 7th, 8th, 9th month. before the fetus dies in that development process. In fact, 18 can be born. Their typical life expectancy is about a year because they didn't develop properly. All right? A trisomy that can occur is trisomy 21, in which the offspring survives, and this is known as Down syndrome. And I'm pretty sure you're familiar with people who have Down syndrome. Because it is a polygenic trait, we can see a wide variety in their mental capabilities, from never beyond a 5-year-old to some that have graduated college. All right? If it is a trisomy that occurs on the sex chromosomes, it's not as bad as what we see with the other things. So if we have 3 X's, that individual typically they are female. They can have some infertility issues, but that's really the only issues that they have. Another one is if an individual is XYY, they can produce a little more testosterone than people who are just XY, but that's about it. The other one is called XXY, and that individual has Klinefelter syndrome. In Klinefelter syndrome, that individual is male, they're typically infertile, and they can develop some secondary sex characteristics of females, meaning breast development. All right? So, let's look at changes in chromosomal structure. So here, the chromosome is broken, and it can be put back together incorrectly. So one case is just what you see here. This is a deletion. So here, this section got broken, and then when the chromosome was put back together, well, it was put back together without that section in there. So this is a loss of a fragment of a chromosome. Another is a duplication, and a duplication is a repetition of a fragment on a chromosome. So we can see that here. So this can occur 2 ways. You can have a problem with DNA replication, where you just make that same copy over again, or this piece came from the homologous chromosome to it. Next is inversion. So you have 2 breaks that occur, and that piece is put back in reverse direction. And lastly is a translocation. So 2 chromosomes are broken, and then these pieces are put onto the wrong chromosome. So once again, they're put back together incorrectly. This is actually showing what is known as a Philadelphia chromosome. So this leads to a type of leukemia where there's a break in chromosome 9 and 22, and those pieces are put on to the wrong chromosome.